Hello. Today we're going to talk about organelles, specifically how organelles work together to maintain homeostasis. Homeostasis refers to the way that your body keeps its conditions relatively constant, kind of like how you always have the same temperature or the same pH or the same blood pressure. Whenever your heart rate raises, your body works to get it lowered. Whenever your pH lowers, your body works to raise it back up. So here is an animal cell, and it's kind of overwhelming. Lots of things going on here. We can split it into three different parts. The nucleus, this big thing in the center right there. The plasma membrane, that's like the outer white circle that's holding it all in. And then the cytoplasm. The cytoplasm is the gel-like substance that all of these organelles are sitting in. So first, let's talk about organelles that store, clean up, and support, kind of like the housekeeping organelles. Cytoplasm, it's a jelly-like fluid, and its function is to hold the organelles in place and basically fill out the cell. And this image is an animal cell where we can see the nucleus and the cell membrane, and all of that tan stuff is cytoplasm. You can't see any other organelles in this view. A vacuole is a large sac-like membrane, and its function is to store materials like water or salt. Animal cells have multiple small vacuoles. We can see like that tiny blue structure. And then this is a plant cell. Plants typically have one large central vacuole that stores their water. Here is a real life image of a plant central vacuole, and there's one of the smaller vacuoles inside of an animal cell, and there's another one. A vesicle, it has a really similar structure to a vacuole, but it's like a tiny little sac, and its function is to transport materials. So here we can see there's a little blue substance, and part of the cell membrane actually pinches off and forms a vesicle, and then the vesicle will transport that wherever it needs to go. Like it could be taking it to an organelle within the cell. Here is a real life picture of a vesicle doing the exact opposite. So if you can picture, this vesicle came up and fused with the cell membrane, and now it's releasing its contents to the outside of the cell. So vesicle, it's like a little vehicle. It moves things around. Lysosome, think Lysol, Lysol cleaner. These are also membrane sacs, but they contain enzymes, specifically acid hydrolases, and they do digestion. So if there's something that we don't need in the cell anymore, a lysosome will engulf it and then break it down. So over here, these orange circles are the lysosome, and the green area is the material being digested. Peroxisome is similar to a lysosome, but it contains different enzymes, specifically catalase and oxidase, and its function is to detoxify toxic substances. So a lysosome could break down anything old, like it doesn't have to be toxic, just like an old cell part. Peroxisomes are specifically for toxic substances. For example, catalase breaks down hydrogen peroxide. The cytoskeleton is like a network of rods or tubes. You can kind of think of like if you can see the tubes or like the beams that hold up a house, that's like the cytoskeleton. And they run all through the cytoplasm and basically hold it up and give it a structure. If you remove the cytoskeleton, it would collapse into like a flat, gooey puddle. So the function is to support the cell, keep it open, and some organelles can help move on the cytoskeleton as well. There's three different types of cytoskeleton. Here's a picture of the three types. We have microfilaments, intermediate filaments, and microtubules. We're not going to really focus on the differences, just wanted to show you them because it's a cool picture. And again, we're picturing them running through the cell, holding the cell up. Centrioles. Centrioles are these little cylinders. They remind me of like Twizzler bites, and they always are found in pairs of two, and they function during cell division. They help organize the chromosomes and make sure they line up so that each cell gets the correct number of chromosomes during division. Now let's focus on the organelles that build proteins. We have the nucleus. It's famous because it's the largest organelle and it contains the DNA. Its function is to control the cell. It's responsible for organizing cell division, like passing on information, and for providing the instructions to make proteins. So, super important organelle. The outside of the nucleus has a nuclear envelope. It's a double membrane structure and it has pores. So see all those little teeny orange things? Those are holes so that things can actually move into and out of the nucleus. Its function is to separate the nucleoplasm from the cytoplasm. So we're keeping the goo inside the nucleus separate from the goo that's the cytoplasm. And the very center of the nucleus is called the nucleolus, and it's made of ribosomal RNA and proteins. Its function is to make ribosomes. Here's all of that together. So the large purple part we would call the nucleus. Here you can see the nuclear membrane, and it's an easier view to see how it's two layers, and there's a little orange pore that would let stuff in and out. And here's the center nucleolus that makes ribosomes. Ribosomes are little tiny particles. They have two subunits, and they're made of ribosomal RNA and protein. When I say two subunits, I mean two parts. And their function is to make proteins. So these little things, you plop an amino acid into them, and then a protein comes out the other side. So the function of a ribosome is to literally like make the protein. Rough endoplasmic reticulum, we can abbreviate it RER. It's like a whole system of membranes. So instead of being a little sac, it's a whole bunch of membranes that coil through the cytoplasm. And the rough ER is called rough because it has ribosomes. So it has the little ribosomes all over it. Its function is to attach a sugar to a protein. So like the ribosome would make the protein, and then it would go to the rough ER, and then the rough ER would attach a protein to it. The rough ER also makes vesicles that are able to transport the protein. The smooth ER is also like a membrane system, but notice that it has no ribosomes. That's why we call it smooth ER. The smooth ER is where we make lipids like fats and cholesterol. It's also where we break down lipids and drugs that were not broken down by the peroxisome. And here's a picture so we can see it all together. So this purple thing, that was our nuclear envelope. So we always find the ER by the nucleus. This part right here that has little orange dots, that's the rough ER covered with ribosomes. And over here is the smooth ER. And this is a really cool electron micrograph where the ER just shows up as like all of these lines. And these little black things are ribosomes. So this is the rough ER. And then over here where there aren't any ribosomes, that's the smooth ER. Golgi apparatus. Golgi apparatus is another stack of membranes. Surprise, surprise. And lots of vesicles. So the Golgi, like little bits of it break off and form vesicles. Its function is to package, modify, and sort proteins so that we can get them ready to be secreted. So the Golgi would basically package the protein and send it out of the cell, or it might send it over to a lysosome to get broken down. And here is a real life picture of what it actually looks like in your cells. Pretty cute. Here's a nice diagram that shows how all of these organelles work together to make proteins. So first, the ribosome actually makes a protein. So there's a ribosome, and then that kind of like little wiggly thing coming out of it is a protein. Some proteins complete their assembly on the rough endoplasmic reticulum. So they're coming through here, they complete their assembly. Remember, the rough ER is going to attach a carbohydrate or a sugar group to the protein. And then proteins are carried to the Golgi apparatus in vesicles. So right here, we can see how the rough ER basically pinches off and forms a little vesicle around the protein, and it carries it over here to the Golgi. Now the vesicle is going to fuse with the Golgi, and then while it's inside of it, it does its packaging and sorting. You can see that it's attached some groups to it right there. And then the membrane is going to pinch off again and form another vesicle so that it can be taken wherever it needs to go. Like this one looks like it's fusing with the cell membrane, so it can be carried somewhere else in the body, whereas this one is just traveling somewhere else within the cell, perhaps to a vacuole. And there we have it all in one. Organelles that capture and release energy. So animal and plant cells both have mitochondria. It looks kind of like this bean shape. And notice that it has two membranes. It has this like outer dark orange one, and then it has this inner lighter orange membrane that's folded into a whole bunch of projections, lots of surface area. The function of the mitochondria is to produce energy. The mitochondria makes a molecule called ATP, and ATP is what provides energy for our cells. And here's another image of a cell. All of these dark orange things are mitochondria, so we can see them everywhere. And here's a different artist's view of it, but we can still see the double membrane system. And the chloroplast. Chloroplasts are only found in plant cells and some bacteria cells. They also have a double membrane structure, but they're like two flat membranes, like 1, 2. And then inside, these looks like little like discs or coins. They're just a whole bunch of like stacks of membranes. So we have membranes inside of membranes inside of membranes. The membranes in here hold chlorophyll, which makes them look dark green. And the function of a chloroplast is to do photosynthesis. So that means it's able to capture sunlight, energy in the form of sunlight, and turn it into glucose. And in this image, we see these dark green things are the chloroplasts. Finally, we're going to talk about organelles that form cellular boundaries. Starting with the cell membrane. The cell membrane is made of lipids. So like one circle right there with the two little legs coming out, that's one phospholipid. And notice there's 1, 2 rows of them. So that's where we get a double layer of phospholipids. And the function is to control what moves into and out of the cell. This is called selective permeability. The head is hydrophilic, which means it can mix with water. These tails right here are lipids. They're nonpolar. They can't mix with water. So if something like water tried to enter the cell membrane, it could go right here by the heads, but it would not be able to go any farther. So that's how the cell membrane controls what can go in and out. Here is another view of the cell membrane in a little bit more detail. So here we can see our same phospholipids, but there's also proteins in the cell membrane. So something nonpolar that would not be able to go through the membrane part might be able to go through a protein. Notice there's some other things like cholesterol inside the cell membrane and carbohydrates on the outside of the cell membrane. Here's a nice view of cytoskeleton, all those purple strands. And the cell wall. The cell wall is a strong supporting layer that's outside of the membrane. It's not found in animal cells, so only found in plant and bacteria cells. And its function is to support and shape the cell and to protect it. So this diagram is telling us that all cells have a cell membrane. All prokaryotes and eukaryotes have a cell membrane. But notice that extra dark piece around it right there is the cell wall around the plant, and the dark piece right here is the cell wall around the bacteria. So only eukaryotic plants and prokaryotic bacteria have cell walls. And that's a wrap. You can take a minute and use this picture to review, and feel free to email me whatever questions you have. Thanks.